my pleasure to introduce our speaker for our seminar seminar series examining dementia across the public health spectrum. Right, so it's my pleasure to introduce Dr. Jill Barnes for today. She's an assistant professor in the Department of Kinesiology at the University of Wisconsin at Madison. She has an affiliate faculty appointment in the Division of Geriatrics and Gerontology in the Department of Medicine at the Wisconsin School of Medicine and Public Health. Dr. Barnes received her undergraduate degree at the University of Michigan, her master's and PhD from the University of Texas at Austin, and she completed a postdoctoral fellowship in integrative physiology at the Mayo Clinic. Dr. Barnes's primary line of research is to understand the effect of aging on the structure and function of the blood vessels in humans and how this relates to the risk of cardiovascular disease and dementia. Her laboratory focuses on how age-associated changes in the structure and function of blood vessels influence the control of blood flow to the brain in humans. These projects are funded through the National Institutes of Health, Department of Defense, American Heart Association, and the Alzheimer's Association. So welcome, Dr. Barnes. The title of her talk today is The Impact of Aging on Cerebral Blood Flow Regulation in Humans. Take it away.
Thank you, Loretta, and thank you for the invitation to speak to you today and to share some of the work that we've been doing over the last few years. So I wanted to start with kind of an overview of the lab and what we focus on. And so our overall research interest is actually in the neurovascular control of the circulation, and in particular, thinking about the large central arteries, as well as the arteries that are supplying the brain, and then the intracranial arteries within the brain. In particular, we're interested in how these blood vessels relate to blood flow in the brain, and then why blood flow starts to decrease as we go through the aging process. So two of the things that I'm going to be talking about today, two of the variables that we measure are vasoreactivity and neurovascular coupling. So vasoreactivity, we have an increase in, for example, arterial CO2 or hypercapnia, and then we measure the cerebral blood flow response and see how reactive those blood vessels are to CO2. We can also measure neurovascular coupling in the brain by having an acute cognitive task shown in these little green bars, and then again measuring the cerebral blood flow response. So why we're interested in these variables and the large central arteries is because there's data from large epidemiological studies, as well as some work from our lab that suggests that these factors are related to neuropathology. This could include gray matter atrophy, changes in white matter health, and decreases in cortical thickness. And as many of you are aware, these changes in neuropathology are linked to cognitive decline. So throughout the last decade or so, there's been a few key questions that connect a lot of the studies that I'm going to be talking about today. So the first is determining how neurovascular control and vascular function change with age. And today I'm going to be focusing particularly on the blood vessels supplying the brain, as well as blood vessels in the brain. Next, understanding how these factors might influence brain health. And then lastly, explore some of the factors that influence cerebrovascular function as a whole. Most of the work will focus on aging, but when we are considering aging, there are other factors that need to also be considered. So I want to briefly review our current understanding of vascular structure and function. So this is the proximal aorta up here, and then the distal aorta. So the proximal aorta is highly elastic. and responsible for dampening the pulsatile blood flow that comes from the left ventricle. So if we compare the structure of the proximal aorta in a 20-year-old versus a 70-year-old, there are some substantial differences in structure. So for example, there's endothelial hyperplasia in an older adult. There is decreases in elastin and increases in collagen fibers. There's also an increase in the vascular smooth muscle cells. And these structural factors can influence the function of the blood vessels. So we can measure this in the large arteries by quantifying pulse wave velocity or quantifying other arterial stiffness measures. In the smaller blood vessels, we can look at pulsatility, which I'll talk about today. We can look at flow-mediated dilation of those vessels, and then we can look at vascular reactivity or vasoreactivity, which I'll talk a lot about today. So the idea is these changes in the large central arteries, what is the impact on brain health? Well, if we look at the pressure profile moving from the left ventricle with those high pressure fluctuations into the aorta and then into the large arteries, the small arteries, the arterials and the capillaries, we see this decrease in pulsatility at the arterials and decrease in pressure overall before reaching the delicate capillaries. If we look at a participant who has elevated arterial stiffness and changes in the structure of those large central arteries, we see those same pressure fluctuations, but they're augmented compared to a normal healthy condition. But you'll also notice that these pressure fluctuations move through the arterioles and maybe even into the capillaries. So there's this high pulsatile flow and high pulsatile pressure that's reaching these delicate capillaries. And the brain is a high flow organ. And so this level of pulsatility in the brain is hypothesized to cause damage within the neuronal tissue. So for example, this increase in pulsatility might manifest as end organ damage, and that can be white matter hyperintensities, it can be cortical atrophy, and perhaps alterations in clearance of proteins such as beta amyloid. Now, we looked at this to see if there was an association between arterial stiffness and cerebral pulsatility, because most of this work is based on either large epidemiological studies that look at these relationships, but they often include adults that have a lot of cardiovascular comorbidities. So we evaluated arterial stiffness in the larger arteries, specifically the aorta, using pulse wave velocity. We had young and older healthy adults, and we evaluated intracranial cerebral artery pulsatility using 4D flow MRI. I'll talk a little bit more about 4D flow MRI near the end of the talk, but this is one of the images that we can get, and it's showing the major intracranial arteries in the brain, such as the internal carotid arteries, the circle of Willis, the middle cerebral arteries, etc. And we can evaluate pulsatility by understanding how much that flow is changing within a given cardiac cycle and quantify that. So we first looked at this potential association between arterial stiffness and pulsatility within the middle cerebral artery that's shown here and the basilar artery, which is shown here. And we did not see any association in young healthy adults. However, when we looked at older healthy adults, we did see a significant and positive association, such that even in an older healthy adult, in the absence of any cardiovascular disease or hypertension, increases in or higher levels of pulse wave velocity, indicating greater arterial stiffness, was associated with higher intracranial cerebral pulsatility. So again, many of these studies that have looked at arterial stiffness and different factors related to brain structure and brain function have been large epidemiological studies with elderly adults and comorbidities. This is a study that was done when I was a graduate student at the University of Texas in Hiro Tanaka's lab, where we looked at arterial stiffness in the large central arteries, again with pulse wave velocity. These were middle-aged adults and otherwise healthy. We looked at cerebral perfusion using arterial spin labeling. And so what we did is we took these middle-aged adults, and we split them between two groups, one with low arterial stiffness, shown in blue, and then high arterial stiffness, shown in red. And indeed, those adults who had higher arterial stiffness within that middle-age range showed lower levels of cerebral perfusion in several key areas of the brain, including the hippocampus. So we've also investigated not just arterial stiffness, but other variables that are associated with arterial stiffness and arterial hemodynamics and brain structure. So we did this in a group of postmenopausal women who had low cardiovascular risk. They were all normotensive, and we looked at white matter hyperintensities. So these white matter hyperintensities are shown here, these bright white areas that indicate demyelination and axonal loss, and these white matter hyperintensities are linked to cognitive impairment. So what we saw in this group of healthy postmenopausal women, that higher aortic blood pressure measurements and higher both systolic and diastolic, as well as higher augmentation index, an indice of arterial stiffness, were all associated with greater white matter hyperintensities. So we also looked at aortic transit time. In this case, shorter aortic transit time indicates a faster velocity of wave travel, which is indicative of arterial stiffness. So in this case, those that had lower aortic transit times had higher white matter hyperintensities. So collectively, this study shows that arterial hemodynamics that are indicative of elevated arterial stiffness were associated with white matter hyperintensities. And we weren't necessarily surprised by this, but the unique aspect of this study is that these women were around the age of 61 years of age, and they overall had very low white matter hyperintensity burden, yet we still see these significant associations with differences in brain structure. So if vascular function declines with age and vascular health can affect brain health, this will most likely affect cognitive decline and the eventual diagnosis of dementia. This is a model from Dr. Constantino Aidicola that is from way back in 2004 that suggested that as we move through the aging process, there's an increase in neuropathology shown by this purple line. So this increase in neuropathology, is really increasing during the prodromal phase of a disease like Alzheimer's disease or dementia. And then once reaching the full-blown disease, neuropathology is extremely high. What's interesting about this model, and this is based on a lot of his work over the last few decades in animal models, that cognitive function, which is shown in the red line, remains fairly high and then starts to decline during the prodromal phase and coincides with this onset of neuropathology. However, his work suggests that vascular regulation, shown in the blue line, declines earlier on in the aging process when cognitive function is normal or unimpaired and neuropathology is relatively low. So we're really interested in this particular area in my laboratory, where we study primarily cognitively unimpaired individuals. Some of them who have, I won't talk about the individuals who have a high risk of Alzheimer's disease, but we do have studies ongoing with those individuals. But we're mainly focusing on unimpaired individuals where we're seeing these slight differences in vascular regulation occurring early on. And our overall hypothesis is that these subclinical early changes might put them on the trajectory of, it might be accelerating either neuropathology or declines in cognitive function. So arterial stiffness, which I had talked about earlier, is one aspect of vascular dysfunction. But whatever method of vascular dysfunction that we're talking about disrupts hemodynamics and disrupts flow. This flow could result in hypoperfusion of the brain or hypoperfusion of certain areas of the brain, but it also could result in hyperperfusion of areas of the brain. And both of these can contribute to neuronal death. In particular, hypoperfusion is often pointed out as a potential cause of reductions in energy substrate delivery, reductions in clearance of proteins such as amyloid, and these can also contribute to neuronal death. And collectively, associated with declines in cognitive test scores. Now, most of the work with vascular function that I've talked about so far is referring to the large central arteries. And we were interested in the ability to quantify vascular dysfunction within the cerebral arteries in humans. So how do we test vascular function in the cerebral vessels? Well, we can look at a control condition where we have normal cerebral blood flow through a blood vessel. We have mean arterial pressure providing some of the perfusion pressure. And then we can expose the participant to hypercapnia or hypoxia or neuronal activity, all of which result in an increase in overall cerebral blood flow. And within a specific cerebral vessel, we would expect dilation as a result of these stimuli. So again, we can quantify this using vasoreactivity, increasing CO2, causing hypercapnia, and measuring the flow response, as well as using neurovascular coupling and utilizing acute cognitive tests to look at the blood flow response. So there's a few other ways we can quantify vascular function. I'll mention one of them at the end of the talk, but most of the things that I'm going to talk about today are focused on vasoreactivity, and only our recent work has now expanded to neurovascular coupling. So we can capture cerebral blood flow a variety of ways. Two of the ways that we use the most is looking at B-mode ultrasound. So this is relying on the extracranial arteries, so the internal carotid artery or the vertebral arteries, where we can get the blood vessel, we can get the diameter of that blood vessel, and then we can get the velocity response and quantify overall blood flow through that blood vessel. We can also use transcranial Doppler to look at intracranial arteries. So we do not get the diameter in this case. The only thing that we get is the velocity of flow through the blood vessel. But we can do this in the middle cerebral artery, the posterior cerebral artery, or the basilar artery. So moving on to talk about some of the factors that influence cerebrovascular function, I mentioned that the majority of the talk is going to be focused on aging, but there are other things that may affect this age-related response or the age-related differences in cerebrovascular function, and I'll show some data involving exercise, also biological sex, menopause age or age at menopause, menopausal hormone therapy, and pregnancy history. So starting with aging, our first study was interested in looking at cerebral vasoreactivity in aging. We utilized transcranial Doppler to look at the middle cerebral artery. So this is showing the middle cerebral artery velocity, and this is showing end tidal carbon dioxide, and we do several different levels of carbon dioxide through the breathing protocol. You can see that the older adults have lower cerebral artery velocity at baseline and at each stage of the protocol compared with the younger healthy adults. If we quantify the slope of this line and we use that to indicate vasoreactivity, older adults had lower cerebral vasoreactivity when compared with the young adults. So this is one way that we often use to look at cerebral vasoreactivity, utilizing several levels of carbon dioxide throughout a breathing protocol and then quantifying that response. So in a follow-up study, we were curious as to whether the cerebrovascular response to acute cognitive challenge was also reduced in older healthy adults. So we again recruited young and older healthy adults, and they performed two different sets of cognitive tests while fully instrumented to measure their hemodynamic responses. So we used the NDEC working memory test, and we also used the Stroop test of executive function. So now these figures look slightly different. We have the young and the older adults. This is showing the magnitude of change in middle cerebral artery velocity during that cognitive challenge. So you can see the young adults here, the individual data is plotted here, and a little bit more variability in the older adults, but there was no difference between the young and older adults in response to a test of working memory. When we looked at the Stroop test of executive function, there was a significantly different response in the older adults. they actually showed a greater magnitude of change in middle cerebral artery velocity during the Stroop test when compared to the young adults. So again, these participants were fully instrumented while we were doing these cognitive tests. So they're not the typical cognitive test setup that you might see when thinking about mild cognitive impairment or dementia. And we were quite surprised because this was against what we hypothesized, because we hypothesized a lower, and we did see an augmented response in the healthy older adults, at least during the executive function test. So now going back to vasoreactivity, where we did see a blunted response in the older adults, we wondered what the role of exercise was. We know that exercise is good for the brain, but trying to understand why exercise might be good for the brain and what it actually does is kind of a core research question in my laboratory. So when we looked at a group of young adults who are untrained or recreationally active, we evaluated their cardiorespiratory fitness using a VO2 max test, and we evaluated their cerebral vasoreactivity shown on the y-axis, and we didn't see any association in young healthy adults. But we knew that the different types of exercise that some of these young participants were doing might elicit differences in VO2 max and have different effects on VO2 max. So we wanted to look at mode of exercise a little bit more closely in these young adults. So the other rationale for this is that previous studies have suggested that arterial stiffness changes as a result of exercise training. For example, aerobic-based exercise has shown to reduce arterial stiffness, but high intensity resistance type exercise has been shown to increase arterial stiffness. And so we realized that the mode of exercise might have an influence on not only the large arteries, but also the vasoreactivity measurements. So for this study, this was a cross-sectional study. We looked at young adults and we recruited them from 3 separate groups. They were either untrained, they were aerobically trained, or they were resistance trained for at least 1 year, but most of them had years of experience and were often competitive. We also had these adults, they were age and sex matched, and we evaluated their cerebral vasoreactivity. So this is a similar figure that we looked at before in the 3 groups. And if we plot these slopes as a bar graph and also showing the individual data, you can see the vasoreactivity between the 3 groups. So there was no difference in cerebral vasoreactivity in the 3 groups, suggesting no influence of the mode of exercise. But you can also see the wide variability, particularly in these training groups, the aerobic trained here and the resistance trained here. So what about the role of exercise and vasoreactivity in older adults? Well, again, in our earlier study where we had untrained and recreationally active adults and we evaluated VO2 max, we did see a significant and positive association between fitness and vasoreactivity, at least in these older healthy adults. But this is just a correlation in quite a small sample size. And none of these adults were exercise trained or aerobically trained. And so we wanted to follow this up with an additional study in a larger sample. So to evaluate this, we specifically recruited habitual exercisers to evaluate the age-associated difference. So there's about 72 participants who were in this study. Many were endurance trained or competitive endurance athletes. And if we look at these vasoreactivity plots, you can see that not too much of a difference between these groups. In fact, there's no baseline differences between these groups as we had seen in the previous study. And when we plot the slope of this relationship, there was again no difference between these the young and older adults. So this is actually, we initially took this as good news because in these habitual exercisers, we did not report any age-associated differences between these groups, meaning that exercise might be have a positive effect on cerebral vasoreactivity. But we did also notice that when we were characterizing the blood pressure response to the increases in CO2, which is a physiological stressor, we noticed that the older adults had significantly higher mean arterial pressure at baseline and then at each stage of this protocol. And so when we calculated the reactivity of mean arterial pressure and the slopes of these relationships, you can see that the older adults had significantly higher reactivity in terms of pressure. So their pressure was not only starting higher, but it was also increasing more during the protocol. So this was a bit of concern because in these two groups, there were no differences in baseline brachial blood pressures, and there were no differences between these two groups at the start of the experimental study day. But when we got to the point where we were getting ready to do the breathing test, you can see that pressure was higher in these older adults. So diving into this particular study a little bit more and thinking about the interpretation, in these habitual exercisers, we don't see age-related reductions in cerebral vasoreactivity. So it could be that there's a beneficial effect of habitual exercise. However, when looking closer at the participants' cardiometabolic profiles, their BMIs, it may have been that these older trained adults had overall better health. Even though in all the studies that we're doing, they're all considered healthy. But in this group, they were particularly, I won't use the term super agers necessarily, but they were very healthy older adults. And so it may not necessarily just be exercise. It could just be that they had better overall health. And so we didn't see the age related reductions or age related differences in cerebral vasoreactivity. So there's also support from the literature on this. So this is our data. I just have it plotted a little bit different way with the different levels of CO2. The young adults shown in black, the older adults shown in white. And this is a study from Coverdale et al. That used one level of hypercapnia, and they plotted their data very similar to the way that we did so that we could compare these. And they also did not see age associated differences in an active cohort of young active and older active adults. So that made us feel a little bit better about the data that there is support from the literature seeing similar results. And interestingly, in this Coverdale paper, they also reported an augmented blood pressure response to the stimulus in these healthy older adults. So when we think about the effect of aging, we often have to consider looking at differences between men and women, looking at differences between males and females as they go through the aging process. So this is a third factor that I wanted to talk about, was the potential for sex differences. So before we dive into vasoreactivity measurements, I wanted to show some data from a different group that was utilizing arterial spin labeling and looking at the difference in cerebral perfusion in young adults, in middle-aged adults, and then in older adults. The areas that's more red show is less blood flow. The areas in yellow is higher levels of blood flow. And in particular, this study showed that baseline cerebral perfusion is less in older healthy adults when compared with young and when compared with middle-aged adults. But our concern is that these potential age-associated differences might not be equal between men and women. And so this is a study by Liu et al. Using arterial spin labeling, as well as our own data looking at cerebral perfusion with arterial spin labeling, looking at young participants and older participants. In both of these studies, young women show higher levels of cerebral perfusion when compared with young men. Same thing in our data, and the young women are the only ones that are showing significant age-associated declines in cerebral perfusion, and our data shows the same thing. So this suggests that any age-associated differences might be more prevalent in women when we're referring to cerebral perfusion when compared with men. But this is just baseline cerebral perfusion at rest. How does that relate to vasoreactivity? Well, looking at sex differences in vasoreactivity, if we look back in the literature, this is a study by Kastrup et al. So we know there's differences in global cerebral perfusion, but what about vasoreactivity? So this is showing their measure of vasoreactivity in men and women. Women are shown in white, men are shown in the black bars through the different decades of life. If you look at the third and fourth decade of life, you'll notice that this study suggests that women have higher cerebral vasoreactivity compared with men during the third and fourth decades of life. and then no age-associated differences after that. Now, because of these differences in global cerebral perfusion, when we calculate our vasoreactivity measurements, the one I've shown you before, as well as the ones that I'm going to show moving forward, is we always account for these potential baseline differences in cerebral perfusion. So we look at the magnitude of change when evaluating our vasoreactivity measurements rather than the overall level of cerebral blood flow or cerebral blood velocity. So in our data, when we look at our young and older healthy adults and we split them up by men and women, we see a little bit different response. So we see that young men actually have the highest level of vasoreactivity compared with young women. So these are significantly different. And then when we move into the older age categories, we don't see any differences between men and women in the older age categories. We also see that all of these groups are significantly lower than younger men. So our data suggests that, at least in terms of vasoreactivity, that it's the younger men who are experiencing the age-associated declines, or in this case, differences between young men and older men, whereas there's no difference between the young women and the older women. So these age-associated differences may not be the same depending on if we're concerned with men or if we're concerned with women. So one of the factors that also may influence either cerebral perfusion or cerebral vasoreactivity is the menopausal process. So this is a fourth potential factor affecting cerebrovascular function, and that's the age at menopause. So not just menopause itself, but what age did a woman go through menopause? So we know that this has a profound influence on cardiovascular and hemodynamic variables, but what about cerebral vasoreactivity? So we evaluated this or investigated this in a group of postmenopausal women who are around the age of 61, and we looked at the age at onset of menopause. So all of these women experienced natural menopause. None of them were taking menopausal hormone therapy. And we just simply split them into two groups, those that experienced an earlier menopause at the average age of 49, and those that experienced a later menopause at the average age of 54. When we evaluated cerebral vasoreactivity, we saw that those that had reached a later menopause, shown in green, and I'm showing the slopes here, had significantly higher cerebral vasoreactivity compared with those that experienced an earlier natural menopause, shown in blue. And this was despite the fact that they were the same match for age and the same age at the time of the study. So women with an earlier age of menopause, if we look at this the other way, women who have an earlier age of menopause demonstrate lower cerebral vasoreactivity measurements. So this brings us to some of the studies that are looking at sex-specific conditions that affect women, because this might influence their risk of overall cognitive decline. So I mentioned age at menopause, but we know that menopause itself and the hormonal shifts are associated with disruptions in cardiovascular risk as well as hemodynamics. But we were interested specifically in cerebrovascular function. And in the studies that I'll present moving forward, it's also concerned with brain structure and brain function. So the first study to evaluate this is we were interested in the effect of menopausal hormones and whether or not menopausal hormones have any influence on cerebral vasoreactivity. So to test this, we had to utilize a clinical trial that evaluated postmenopausal women and recruited them into a clinical trial. So this clinical trial was part of the KEEPS, and the site was under the direction of Dr. Virginia Miller at Mayo Clinic. So we utilized this group of women who had previously participated in a clinical trial to look at the effect of menopausal hormones. So they had previously been randomized to either a placebo group, a transdermal 17-beta estradiol group, or an oral conjugated equine estrogen group. So when they were in the clinical trial, they received this treatment for 4 years, and then they were evaluated 3 years after treatment. So many studies suggest that when a woman is on menopausal hormones, it does affect cerebral vasoreactivity, but we were interested in whether or not there was any sustained effect of menopausal hormone therapy given for a relatively short duration with a washout period. And so this is the cerebral vasoreactivity measurements. The individual data is shown with then the gray bars indicating the averages for the group. So this is the placebo group, the transdermal 17 beta estradiol group, and then the oral conjugated equine estrogen group. So there was no significant differences between these three groups 3 years after treatment. And if we combined these two groups, the two groups that actually received the active menopausal hormone therapy, there was only a marginal sustained effect. So overall, we concluded that there's really no difference in these groups with cerebral vasoreactivity, which is not necessarily a negative thing. It could also be thought as positive, because that means if a woman decides to take menopausal hormone therapy, that there's not necessarily an effect 3 years after the fact. So the other thing that we were interested in this study was looking at brain atrophy. And in this case, we were lucky enough to have scans of these participants at the beginning of the trial, as well as throughout the trial, and then the 3 years after. So this is showing brain volumes, and this is showing ventricular volumes within these three groups. So in this case, the gray line is showing the placebo group, the red line is showing the transdermal estradiol group, and the yellow line is showing the oral conjugated equine estrogen group. So this kind of grayed out area is showing, we have the baseline scan, we have the scan at the end of the trial where they were receiving menopausal hormone therapy, and then we have the scan that was conducted 3 years after. So what we can conclude from this study, even though there might be slight differences immediately at the cessation of menopausal hormone therapy, overall, there was no group differences in rates of brain atrophy between these three groups. So this suggests that the rate of atrophy were not different over the 7 years of measurement. So the last factor that might influence cerebrovascular function that I'll talk about today is pregnancy. So there's been studies that looked at cerebral vasoreactivity and cerebral blood flow during pregnancy, but also in the immediate postpartum period. We were interested in the influence of pregnancy history after menopause and how that affects cerebrovascular function, because after menopause is when women have an increase in their cardiovascular risk as well as stroke risk. So we compared women who had a history of a normal pregnancy versus women who had a history of a hypertensive pregnancy, and specifically preeclampsia. So these were postmenopausal women. This was a case-controlled study based on pregnancy history, and we evaluated them 35 years later. So in order for us to do this, we relied on the Rochester Epidemiology Project to be able to go through medical records to recruit these participants into the study. So these were postmenopausal women. We were testing them 35 years after the incident pregnancy. This is showing, again, the end-tidal carbon dioxide and the middle cerebral artery velocity. The blue line is showing the women who had a history of normotensive pregnancy. The red line is showing the women who had a history of preeclampsia. So these individuals, these women who had a history of preeclampsia demonstrated lower middle cerebral artery velocity at baseline, and then they also had lower vasoreactivity compared with the women who had a history of normotensive pregnancy. So this effect remained significant even after adjusting for any potential differences in blood pressure as well as BMI. So vasoreactivity is reduced in women with a history of preeclampsia, even if we're talking about 35 years after the incident pregnancy. So because preeclampsia is characterized by the excessive increases in blood pressure during pregnancy, we were interested in whether or not these women with a history of preeclampsia demonstrated an altered cerebrovascular response to a hypertensive stimulus. To investigate this, we compared the response during isometric hand grip. So isometric hand grip is known to increase blood pressure. It's often used to characterize cardiovascular reactivity and potential risk of cardiovascular disease. But we utilized it to look at how the cerebral circulation was responding to this hypertensive stimulus. So this is showing baseline, and then we have 3 levels of isometric hand grip. Then we inflate a cuff immediately after exercise, suprasystolic, and then we allowed them to recover. So these first 2 lines are showing the change in mean arterial pressure during this isometric hand grip and then in the post-exercise occlusion phase. So the blue line showing those with normotensive pregnancy, the red line showing those with a history of preeclampsia. You can see that the blood pressure responses are very similar. However, when we looked at the change in middle cerebral artery velocity, which was shown on this axis here, we see the red line for those with a history of preeclampsia and the blue line, those with a history of normotensive pregnancy. And there were statistically significant differences between groups. That third portion of exercise, the latest phase of exercise when blood pressure was highest. And we also saw significant differences even in the recovery phase, despite the fact that the blood pressure levels were quite similar between groups. So this suggests that the women who had a history of preeclampsia, their blood velocity was continuing to increase when in an ideal situation, we would have more of a flat-lined response like those who had a history of normotensive pregnancy. So this suggests that pregnancy history, even when tested 35 years later, may impact the cerebrovascular response to a hypertensive stimulus. So it's showing us that there's something different about the cerebral blood flow regulation that's occurring in these women who had a history of preeclampsia. So we also looked at the women who had a history of preeclampsia and split them into groups that were currently hypertensive or normotensive, and looked at their overall brain atrophy that was occurring and saw some interesting results. So I thought I would include it here. So when we evaluated women who had a history of preeclampsia and current hypertension, they had lower gray matter volume in several key areas of the brain, the occipital cortex shown here, as well as the prefrontal cortex and the sensory motor cortex. So collectively, these studies on the women who had a history of hypertensive pregnancy suggest that we have a limited understanding of how such sex-specific conditions might be affecting not only the cerebral vasculature, but also overall brain health. So to summarize these studies on pregnancy history, we have the woman who goes through a normotensive pregnancy and then hits menopause, compared with a woman who has a history of preeclampsia and then also hits menopause. And I didn't show any data on vascular activation, but we do have data that suggests altered levels of vascular activation. I showed some data on reduced neurovascular control of the cerebral circulation. I showed some data on gray matter atrophy, and collectively these could be linked to impairments in cognition in the women who had histories of preeclampsia. So I'd like to wrap up the talk by giving you a few methodological considerations that's kind of informed our most recent work. So I mentioned that most of these studies use ultrasound or transcranial Doppler ultrasound. And the problem is that doesn't allow, the transcranial Doppler ultrasound doesn't allow us to get diameter measurements. So we can't really capture the vessel diameter, and if that vessel diameter is changing, that could influence the interpretation of the data. So with these vasoreactivity measurements, particularly with increases in arterial CO2, these increases in cerebral blood velocity that have been shown, if the diameter is changing, that may not be reflective of what's happening with flow. And so some studies have utilized structural MRI to evaluate the middle cerebral artery diameter in response to CO2. Some of the earlier studies suggesting there's no change in the diameter. So this is the middle cerebral artery in 2 different conditions. But as MRI technologies have gotten better and using 3T or 7T scanners, more recent studies are suggesting that during sustained changes in arterial CO2, that the middle cerebral artery diameter might be changing. So because of that, we followed up some of our studies looking at aging and also looking at sex differences on vasoreactivity using 4D flow MRI. I brought up this picture before where we get velocity profiles and we can get diameter measurements in several large intracranial arteries at rest and during increases in CO2 or any other stimuli that we choose to test. So when we looked at young and older healthy adults and looked at vasoreactivity, we saw an age related difference between these groups when we looked at the whole brain, but we also saw these differences within each of the intracranial arteries that we evaluated. So we have the basilar artery, the left and right internal carotid artery, and then the left and right middle cerebral artery. So this verifies some of the earlier work that we did, but utilizing a different technique that accurately quantifies the flow responses within these individual vessels. So we also looked at the degree of middle cerebral artery dilation between young and older adults. So we do this during the vasoreactivity measurement. So this is the left middle cerebral artery. This is showing cross-sectional area in young adults and in older adults. And the individual lines are showing the individual data, with the black lines showing the group average. So when we looked at this and compared the young and older adults in terms of the degree of dilation, we saw that there was significant vasodilation in the middle cerebral artery only in young adults. And this is likely due to this large amount of variability that was noted in the older adults, with some groups not changing, some individuals decreasing, and some individuals increasing. We also utilize this technique to look at the effect of age, as well as potential sex differences between young and older adults. So this is showing vasoreactivity again in these 5 intracranial vessels: the basilar artery, left and right internal carotid artery, and the left and right middle cerebral artery. And this shows very similar to the results that we saw with transcranial Doppler, where the young men shown in the black bars had higher vasoreactivity in all of the vessels when compared with the younger women. So there's sex differences between young men and young women. And then the majority of the age associated difference was only occurring in men. If we took a closer look at this left middle cerebral artery and we looked at the degree of dilation, we see that there's significant dilation in the men, where we don't see much dilation of the left middle cerebral artery in response to increases in CO2 in the women. So verifying our earlier data with age-related differences only in men, but now we have new data on cerebral vasodilation that's primarily occurring in the young men, with little to no dilation occurring in young women in response to CO2. So to summarize some of the work that we talked about today, one of the questions was, how does cerebral vascular physiology change with age? And so most of the studies suggest that even in healthy older adults, cerebral vasoreactivity is blunted with age. And I showed a little bit of data on neurovascular coupling, and that in our hands is either unchanged or enhanced with age. However, the age-associated decline in vasoreactivity might be sex-specific. Our data suggests that age-associated decline is primarily happening in men, if we're referring to vasoreactivity, whereas others have shown, as well as our preliminary data, that declines in cerebral perfusion may also be sex-specific, but occurring primarily in women. And these conclusions might be influenced by some of the methodologies that are used to quantify cerebrovascular function. How might this influence brain health? Well, I showed data showing associations between arterial stiffness, cerebral perfusion, cerebral pulsatility, and white matter hyperintensities, but there's a lot more work that's necessary in this area. And we did see, at least in the women who had a preeclampsia, that the differences in vasoreactivity did correspond with lower brain volumes. But how the relationship between vasoreactivity, cerebral vessel function, as well as neuropathology is unclear at this point. So we talked about some factors that influence cerebrovascular physiology. Exercise might be one that we would hope might slow age-related changes, but we need to understand more about what's happening. There's a lot of questions, and we don't know the mechanisms if it is slowing the age-related changes in cerebrovascular physiology. This may also be sex-specific. And then I also talked about sex-specific differences, as well as sex-specific conditions such as pregnancy and menopause. And the cerebral circulation in humans really presents a unique set of challenges to try to assess cerebrovascular function and understand what's happening. So we've been trying to use multiple imaging modalities to approach our research questions, as well as collecting additional variables, utilizing different physiological stressors to try to comprehensively understand potential changes with age and potential sex differences. So with that, I'd like to acknowledge the collaborators for the studies that I've shown today, as well as the students in my laboratory who worked on these studies, and then our entire group, as well as the funding sources. And I'd be happy to take any questions at this point. Thank you for your attention.
Jill, thank you so much. That was incredible. If people have questions, I don't know, you can either put a question in the chat. Rachel Yaffe says, thank you. While people are formulating a question, I'll start with one. Do you ever add a cognitive task to your experimental protocols? I'm just wondering if, you know, some of the slopes that you presented for the various hemodynamic outcomes would correlate with executive function scores or memory scores, while you're simultaneously observing the changes in reactivity or blood flow, et cetera.
Yeah, that's an excellent question. So when we're doing the cognitive tests, we don't quantify or score at that time, because the way that they're instrumented in things, there's just a lot of things happening, and I would argue that it may not be correct. However, in our studies that we have currently moving forward, we do have comprehensive cognitive test scores that are available to us. We don't perform that. That's performed by our neuropsychology colleagues, but we would have that information to see if, for example, someone who has, all of them are cognitively unimpaired, but perhaps someone who has a little bit lower scores in executive function, if they have, like, what the magnitude of their hemodynamic response might be when we assess a test of executive function within the lab. We don't have that data, to be honest. We haven't even thought to look at it. So that's a great suggestion of trying to understand these subclinical differences in different cognitive domains and how that might relate to some of our hemodynamic variables.
Right, it might be too early.
Yeah, exactly.
Right, it might be just too early, and maybe these hemodynamic changes happen much sooner than the change in actual cognitive function.
Yeah, and I would agree with you. So that's kind of why we feel like these associations that were, you know, we hypothesized originally years ago to see additional associations between some of these factors, but I think the temporal relationship is off. So the only way we're going to be able to see it is, and we are doing that with some of our current studies where the participants are involved in a longitudinal cohort. So we will be able to follow them up. And then the other thing I was going to mention about the hemodynamic changes is we, just as a experimental design, we do familiarize the participants on a different day with all of these procedures, so that, for example, the day of, it's not the first time they're experiencing the situation, so that we're not capturing, you know, some increase in stress that might be occurring because of the not being familiar with the experimental setup.
Got it. Well, I can appreciate now the role of early menopause on vascular reactivity and brain health. Okay, we've got a couple of questions here. This is from Jerry France. Could you please go over again the differences you mentioned between aerobic training versus resistance training?
Yeah, so differences in arterial stiffness. Well, I'll start with that. So there's been data even in younger healthy adults that arterial stiffness, we assume exercise is going to be result in an improvement in vascular function. But that data suggests that when we're talking about arterial stiffness, that high intensity resistance only exercise has been associated with increases in arterial stiffness. This has been shown both acutely over one bout where it acutely increases and then comes back down. It's been shown with 4 months of resistance training where arterial stiffness increases, and then during 4 months of detraining, it'll come back down. And so because of that, we were interested in looking at cerebral vasoreactivity, hypothesizing that we might see the same thing where resistance training might have a negative impact on the cerebral circulation versus aerobic training. In fact, what we reported with the cerebral vasoreactivity measurements in young healthy adults is we don't see any group differences. We did have adequate power, so it doesn't suggest that it was a power issue. I would also say that because these were young healthy adults, the other thing is they might just be operating on the continuum where their vascular function is already very healthy, and so the amount of exercise they were doing was not going to have a significant impact. So this should be followed up by looking at people who have a longer history of training, such as people in their 40s and 50s who may have been performing aerobic training or resistance training over the course of decades rather than over the course of 5 to 10 years. I'm not sure if that answered the question, so I'm happy to take a follow-up.
Okay, if there is a follow-up here, thank you. I guess you did answer the question. Okay, here's a second question. That was a comment about how informative your talk was, but also what could be the possible reasons behind the early menopause and the related cerebral blood perfusion changes?
Yeah,
I think a number of us are want to know this.
another good question. So I should, in that study, these were healthy women who experienced natural menopause. So we aren't talking about women who experience very early menopause or surgical menopause. So just slight differences in the age at menopause and showing these changes in cerebral vasoreactivity. The other thing that we looked at was the timing of the years since menopause, because the group was similar age when we were studying them. And that didn't seem to have an effect as much as the age at menopause had an effect. So we really don't have an answer as to why that is the case. In fact, we haven't published that work yet. That manuscript is currently in preparation because we are a little bit concerned about that. We want to make sure everything is verified. We wanted to see if we can get additional subjects to see if we can repeat this. So far, it's holding up. But there are slight changes in blood pressure, even if you're looking at normotensive women, when we look at those two groups. So we also had to statistically account for the current level of blood pressure. So I don't know the answer, but my suggestion would be these slight elevations in blood pressure that occur, even if we're talking about 2 millimeters of mercury, might be an underlying factor with the earlier age at menopause. We don't have comprehensive data in terms of the cerebral circulation using 4D flow MRI with these participants yet. And we also don't have comprehensive arterial stiffness measurements or carotid artery measurements in these participants yet. We are analyzing the data right now. So that might give us some additional answers. But my first thought would be that these slight changes in large artery stiffness or slight changes in blood pressure that are associated with menopause might be underlying it. And even though they're only a few years ahead in the menopausal period, that it is having some effect on these cerebrovascular responses. So I don't know if it's something specific in the cerebral vessel or if it's the systemic blood pressure and hemodynamic response that we're capturing.
The bottom line, it's the loss of estrogen and the longer time period of loss of estrogen, I would imagine. Okay, well, thank you. Oh, one more question, Keith Cole. Has the influence of metabolic disease on vascular reactivity been established?
My short answer would be no. There's been a few studies that have looked at obesity with metabolic syndrome. And some studies have, I think there's one study that I'm thinking of that did not see any differences in younger adults who have obesity and metabolic syndrome in terms of the cerebral vasoreactivity. Now, again, if that's done in a middle-aged cohort where perhaps they've had metabolic syndrome over the course of decades rather than an acute period of time, if they're, for example, 30 and have metabolic syndrome. But I don't think there's a lot of data on cardiometabolic disease, metabolic syndrome, and obesity. If we look, there is some data on type 2 diabetics, and they do show differences in cerebral vasoreactivity, but we're also looking at not just age differences, but we're also looking at the cardiometabolic disease over a course of years, because I believe those studies were in their 60s when they evaluated that. So I think there's still a lot of questions as to the time course of when the cerebral vasoreactivity measurements or cerebral vascular function measurements might be changing during the course of disease. I have a colleague who's looking at even kids with insulin resistance. They're looking at cerebral vasoreactivity measurements and trying to understand if they're changing and what factors they're being influenced by. But I think the short answer is no, we don't know enough about it. Probably if we're looking at type 2 diabetics who have had the disease for many years, they are going to show reductions or they're going to show lower levels of cerebral vascular function.
Okay, that's all we have time for. I know everyone is joining me in thanking you for fabulous talk, Jill. Until we meet again.